Hello, dear students, and this is my second lecture on antigen antibody reactions. Now, we have studied about the precipitation, agglutination, or agglutination test, and now we will be going further. So, in this lecture, we are going to discuss about the complement fixation test, neutralization test, opsonization, radioimmunoassay, enzyme immunoassay, chemiluminescence immunoassay, immunochromatographic test, and immunofluorescence. So coming to the complement fixation test. Now, what as the name of the test says, complement fixation. So we are fixing the complement with the antigen and antibody that we are using. That's the principle. And what does it requires? It requires antigen, antibody, complement, sheep erythrocytes, AMBO receptors that are the rapid antibody to sheep red cells. Okay. So this kind of a test is now obsoleted, but then too for theoretical purpose, we have to have a look. So here is a negative CFT. Why it's negative? When complement is added to the sheep erythrocyte coated with AMBO receptors, the lysis takes place. That means it doesn't have any antigen antibody complex. So if lysis is taking place, that means it's a negative test. In the positive test, we have to have an antigen antibody complex which combines with complement. So these complex formed, the complement is fixed by the antigen antibody reaction, and the indicator, the AMBO receptor, the sheep lymphocyte, the indicator system is separated. So lysis does not occur. So that means it is positive. Interpretation: negative CFT lysis of erythrocytes takes place. Complement is not fixed. Serum did not have any antibody. In positive, no lysis, complement fixed. That is, serum contained antibody. There is one indirect complement fixation also. We will not read this this is for not for MBBS. I mean, this is for MD students. So we move on. You just, being an MBBS friend, you just have to know what is CFT, complement fixation test, what is positive, what is negative test. And now we're coming to the neutralization test. Now, the term neutralization itself means to reduce the effect of anything, right? So we are just reducing the effect of toxins, the virus. So there are two kinds. One is virus neutralization test and other one is the toxin neutralization test. So coming to the virus neutralization test, it is defined as neutralization of viruses by their specific antibodies. Is called as virus neutralization test. In which virus we are using? It's virus hemagglutination inhibition test used for diagnosis of influenza, mumps, and measles. Okay. So nothing, the antibodies are neutralizing it. Okay. So when the virus specific antibodies are injected into the system, replication and growth of viruses is inhibited, forming a basis of this test. Now, the second is the toxin neutralization. Again, some biological action of toxin is neutralized on reaction with the specific antibody, that is antitoxin. So it is vivo and vitro. So you should know the examples because you could be asked, like in vivo is example is Schick test for diphtheria. Schick test you'll be learning when you will be reading diphtheria. Okay, and in vitro, inhibition of the toxin effects, like we are adding anti-streptolysin O test for streptococcus virugens. This you can be asked as ASLO is kept in your practical exams also. And Naegler reaction for alpha toxin of Clostridium perfringens. These are some example of toxin utilization test. Now coming to opsonization. Opsin was given by Wright in 1903 to a heat labile substance present in a fresh normal serum, which facilitates phagocytosis. What I call is in one of a physiology textbook it was written that opsonization is a process of making a bacteria tasty for the phagocytotic cells. Okay, so that's what is. Now, this heat stable serum factor with a similar activity is also called as bacteriotropin. And now the term opsonin is now used to refer both these factors. Okay, so there are some indexes to study and all. We don't need to go in further detail. You just have to know that this opsin index is measured by incubating fresh cited blood with the bacterial suspension at 37 degrees Celsius for 15 minutes. And we have to estimate the number of phagocytosed bacteria per polymorph in the blood flame. So that's how we measure this index. And that index is used to study the progress of resistance during the course of disease. So you just have to have some knowledge about the opsonization. Now coming to important topic, that is radioimmunoassay. Now again, this was also previously used, but now it is not being used. Its higher version is being used, but let us have a look on it. First ligand assay method developed by Burson and Rossell Yellow in 1959 to detect and quantify human insulin using anti-human insulin antibody. Okay, and in 77, Yellow also got Nobel Prize for this very sensitive method and used to quantization of substances. And what is the principle? It is based on the competitive binding assay, in which fixed amount of antibody and radio-labeled antigen react in presence of unlabeled antigen, and the labeled and unlabeled antigen competes for the limited binding site on the antibody, and this competition is determined by the level of unlabeled antigen present in the reacting system. After the reaction, the antigen is separated into free and bound fractions and their radioactive count measured. The concentration of test antigen can be calculated from the ratio of bound and total antigen label using standard dose response curve. So this was theoretical. Now let's see this, what's happening. So what is this? The labeled antigen and antibody in the saturated concentration are added. Now we add the patient serum. Now if the antigen in the patient serum are present, after adding protein a precipitation of antigen antibody complex will be formed. And if there is no antigen in patient serum, no precipitation will be formed. Okay. So how we are seeing, how we are plotting, we will be seeing this by a graph that will be between the ratio of bound is to total antigen and the is to concentration of the unlabeled antigen. So what are the uses? It is used in quantization of hormones, drugs, tumor markers, immunoglobulin E, and viral antigen. radioimmunoprecipitation. It is used to measure the antibodies against HBsAg, Coxella burnetii, widely used in clinical chemistry and toxicology and endocrinology. So what are the advantage? It's precision and high sensitivity. It has ease to isotope conjugation. Unlike enzymes, isotopes labeled are not likely to disrupt the antigen activity. Disadvantage: iodine 125 labels reagent in common use have limited shelf life. It is hazardous and it's expensive. That's why we are not using at present this. So what are we using in our labs? We are using enzyme immunoassay. Okay. So it is an immunoassay where estimation of enzyme level antibody or antigen or hapten is done, and enzyme labeled conjugate were first introduced in 1966. Widely used procedure in clinical serology due to their vast versatility, sensitivity, simplicity, economy, and absence of radiation hazards. That's why we have switched from RIA to enzyme immunoassay. Okay, they are basically of two types, that is homogeneous and one is heterogeneous. So what is homogeneous enzyme immunoassay? This is a test which can be completed in one step with all reagents added simultaneously. No need of separate bound and free fractions, and can be used only for assay of hapten such as drugs and not the microbial antigen and antibodies. Example of it is enzyme multiplied immunoassay technique used for small molecules like opiates, cocaine, barbiturates, and amphetamine in serum. So what are the advantages? It is very simple and it has only one step to perform. What are the disadvantages? Microbial antigen and antibody cannot be assayed. So coming to the second type, which is heterogeneous, it involves multiple procedure with the reagents added sequentially and separation of bound and free fraction required either by centrifugation or by absorption on the solid surface and washing. So ELISA, that is enzyme linked immunosorbent assay, is the major type of heterogeneous enzyme immunoassay. So you have to know this. With this, which kind of type of enzyme immunoassay it is? Heterogeneous. Now coming to the detail of this, ELISA, it is a heterogeneous enzyme immunoassay which relies at the immunosorbent, that is and absorbing material that is specific for one of the components of reaction to affect the separation of free and bound fraction. So what's the principle? It is the solid phase immunoassay where one of the reactant, either antigen or antibody, is bound onto solid phase. The sample in which the antigen or antibody has to be detected is added, and if the presence of immune complex, if it is like if antigen or antibody which we are seeing it is present, the immune complex is formed and free antibody and antigen can be removed by washing. The bound antigen antibody complex can be detected using enzyme labeled antigen or antibody. So what are the requirements? We required of solid phase, which is a supporting system, which is to fix the antigen antibody, which can be of polystyrene or polyvinyl or polycarbonate microvels, and it is also made up of beads or tubes or nitrocellulose membrane. And ELISA is done by using the 96 cell microtiter plate. So this kind of a plate is required to perform ELISA, and in this we put the reagents and serum and all. So what a kind of antibodies which we use are enzyme labeled monoclonal antibodies, which is are used because they're highly specific. Now enzymes, some of the enzymes used are horseradish peroxidase, alkaline phosphatase, beta D galactosidase, glucose oxidase, carbonic anhydrase, lysozyme, and etc. Substrate, they should be chromogenic, safe, easy to use. So we have to have a solid base on a plate which we are going to perform. We have to have a antibody that was monoclonal, and we have enzyme, we have substrate. So enzyme for alkaline phosphatase substrate is para-nitrophenol phosphate. For horseradish peroxidase, it is TMB. For beta galactosidase, it is lactose or lactoferrin ceramide. So according to enzyme, substrate are used. So washing solutions, what we take that are buffers, that are phosphate buffered saline, which contains 0.05 to 20, and diluted buffers and blocking agents like PBS or T used in dilute buffers. So that can be sodium casein, non-fat milk, gelatin, newborn, calf, bovine, serum balloon, etc. are the blocking agents. And we need a ELISA reader that is a spectrophotometer. So these are all what we require. Now coming to the ELISA. So what we require was, we required a base, that is a plate that made up of polystyrene or polyvinyl, anything, and or bead or tube or whatever. We require enzymes, we require substrate, we require antibody, we require buffer solutions, we require washing solutions. Okay. And we require a spectrophotometer. So what these are all required for the ELISA. So what are the types? It's direct, indirect, sandwich, competitive, and modified, like IgM capture, which is used in dengue. Okay, and membrane bound cassette ELISA, ELISpot, and biodin avidin linked immunosorbent assay. So what is a direct one? Direct, it is just used to detect the presence of specific antibody in the patient's serum. And what is there? The wells are coated with the antigen. We put the patient's serum that is labeled with enzyme, and we add the substrate, and color develops. If the color develops, the presence of antigen antibody reactions taken place. So specific antibody are present. So main problem to encounter with this test is that the antibodies for detection of each antigen needs to be linked with the enzyme, which makes the test cumbersome. Now, indirect ELISA. What is indirect? In this test, antigen-coated wells in patient serum containing specific antibody which are unlabeled is added to each well. Okay, so what happens is, let's see the diagram. It will be easy to understand. the antigen is coated in the well. We put this tested serum which has antibody in it, that is a primary antibody, and now the enzyme links second antibody which is going to be added, that is the AB2, that is the enzyme linked to second body, that is AB2. Anti-human immunoglobulin antibody are made up of rabbit, sheep, and goat is added. This is here, and then we are going to add a substrate. And if the color comes, then it is positive. So intensity of color is directly proportional to the concentrate of the primary antibody of a patient. Now, third test is the sandwich. What happens in this sandwich? Now, the wells are coated with the antibody. We put the sample, the antigen gets sandwiched between the second antibody, which we will be adding it afterward, and that second antibody is enzyme labeled, and the color is going to be produced, that will be the positive. Now, this again will be the optimal density, is going to measure the color and the amount of antigen. So there will be why it is sandwiched because the antigen is sandwiched between the two antisera, which is raising the two different species against different epitopes of the same antigen. So there is also indirect sandwich, which you don't need to understand. Will come on the competitive ELISA. So in the competitive ELISA, we the antigen are coated in the well, sample is put, and in that antibody are there in the tested sample, and then we add conjugate, we add a specific antibody in the sample which competes with the enzyme level antibody. Okay. So conjugate is washed out as an antigen is not free to bind the conjugate. So if we add substrate, color won't be produced. So what's happening here? Both unknown antibody and antigen. So here both the unknown antibody and the known antibody compete with each other of a fixed amount of antigen immobilized on the wells and on the titer plate. So competitive ELISA yields an inverse curve, that is the amount of hydrolysis of substrate is inversely proportional to the amount of antibody present in the sample. So what is the advantage? Advantage is crude or impure samples that can be analyzed for the presence of antigen in by this technique. Okay. So next type is IgM. In that, what this competition was going on between the unknown antibody and the known antibody. Known antibody, unknown was in the test sample, and known was in the enzyme labeled antibody conjugate. Now IgM capture ELISA, the enzyme immunoassay method for antibody detection, primarily immunoglobulin G. However, specific IgM detection methods can be used to differentiate the recent from the past. You already know that IgM means recent infection because IgM is present early and in the course of most infections. And enzyme immunoassay formats for immunoglobulin M detection used an enzyme labeled anti-immunoglobulin M antibody as conjugate. Here, immunoglobulin G directed against immunoglobulin M is bound to solid phase. Drawbacks of IgM detection by conventional technique: presence of excessive immunoglobulin may give false test, and the presence of auto antibody gives a false test. Use for the congenital infections, detection of IgM in fetal neonatal blood. Okay, indicates active infection. So congenital immediately it can be used, but we also use in the dengue test and all. Now coming to the cassette ELISA. So it is a simple modified ELISA that may be used in a rapid serological diagnosis, and is tested in the cassettes. So what's the principle? It is used to detect antibodies. The specific antigen is spotted onto the nitrocellular paper in a cassette. Beneath the nitrocellular paper, there is absorbing material. The patient's serum is added in a sucked through the nitrocellular paper by absorbing material. And if the serum containing antibodies, they bind to antigen in the nitrocellular paper, enzyme labeled antiglobulin is added to detect the bound antibody, followed by addition of the substrate. So color is produced depending upon the spot. So this cassette ELISA, you will be getting this in your exams too, because in HIV we are using this cassette ELISAs. So what we do is there is a square like cassette present, and on that nitrocellular pad is being placed, and in that the antigens are being coated, and dots are coated, and we put the sera and we put the substrate and enzyme substrate. So color develops, that means antigen antibody reaction has taken place and it is positive. So we are using for HIV, we are using for group A streptococcal focus infection, influenza, chlamydia. We're using the advantage is that it is visual. It is in 10 minutes we can find the result and can be used for in one sera at a time. It has inbuilt controls and does not require washing. The control is also there, and on control there is anti-human antibody present. So that's why. So ELISpot, it is a modification of ELISA which follows the quantitative determination of the number of cells in the population that are producing the particular type of molecule. So what happens is in the capture antibodies are there. It is mainly used for the anti-cytokines antibodies detections and all that. So this is useful for detection of antibody producing plasma cells, cytokine producing T cells or monocyte. In the mix, we have to, if we want to enumerate them. So what happens is that the capture antibodies are coated and the PBNC, and if again non-secretor cells are there and cytokine cells are there, then enzyme labeled antibody anti-mouse antibodies are added, and when we wash, and color spot indicates that the presence of cytokine secreting cells. So this is just for your knowledge, not for detail. As for details, MB students has to require the pathological thing is there, not for the we don't use in this microbiology. There's also one test that is known as the biotin evident linked immunosorbent test, and is an indirect test that can be made for more sensitive by using biotinile antibody this tree and evident complex enzyme for probing. So this also you just for the MBBS students you just need to know that there is a biotin evident immunosorbent test, and which is being used for viral infection, parasitic like toxoplasmosis, kala azar, bacterial infection like tuberculosis, chlamydia, and also for the detection of antigen in viral and parasitic like Cryptosporidium parva. So you should know some examples that this test is used in these, because you don't need to go in detail of every. You just need to go in detail of what the direct ELISA, indirect ELISA, the competitive, then the sandwich ELISA. Okay, so and even this immunoglobulin capture ELISA, these are the ELISAs which you need to go in the detail. Otherwise, the ELISA you should just know the names also and for what they are being used. It's enough for that, because there are multiple kind of ELISAs present in the world, right? So coming to the other technique, that is the chemiluminescence immunoassay, which is being used in biochemistry mostly. And it's nothing that we are just using the light, energy of light for the testing. That is the chemiluminescence refers to the chemical reaction emitting energy in the form of a light. So we are using that, and it is an extension of ELISA where the chromogenic substrate is replaced by luxogenic, that is a light emitting substrate, such as luminol or acridine esters. So that is being used. So the light generates can be quantitatively measured on a luminometer, and this test is 10 times more sensitive than the chromogenic assays. Okay, so you should, in microbiology, you just have to know the name, because we're not using the chemicals. No. Immunochromatographic test is very important test. Now this is used in the case of hepatitis B antigen also, and we are also using it with the HIV. So what happens is, this is a cassette, and this again cassette is coated with the nitrocellulose membrane, and what happens is that we add the sample here, the required amount of sample which is on the brochures. So we have to, according to the antigen and all, we add this serum. And like in hepatitis, in some we require buffers, we don't, in some we don't, but in hepatitis we don't require the buffer, so we just add the serum. And what happens that the gold colloidal particles are here in the in near the serum spot. So what happens, the lateral flow is there. Due to the lateral flow of the serum, the capillary action with the capillary lateral flow, it the takes some gold particles, serum takes some gold particle from here and proceeds to the nitrocellular pad. And here we have some immobilized area and mobilized area. The immobilized area are the C line and the T line. Here in the T line, the if we are taking, we want to see for antigen, antibody will be quoted. And if we are seeing for the antibody, then antigen is going to be quoted. Now, because we are seeing antigen, so antibody is going to be quoted on T, and on C, anti-human antibody are quoted. It is always has to be remembered that the control line will be always after the test line, because as with the capillary action, the serum moves above. We should go to the we should finish the test for that indication. We should touch the control line. If the control line will be in front of the test line, we won't be able to understand that the reaction, the test has occurred fully or not. So you have to know this, that see, it's a negative test in here. Control should always be there. If there is no control present, then it is invalid test, and control should always be there, whether it's negative test, whether it's positive test. If there is positive test, two lines will appear on the control and test, and whether it's negative, the test will appear, there won't be any line of test, but control will, that means it's valid and it's negative. So with the capillary action, the lateral flow, there is the serum goes, and the antigen antibody reactions takes place with the help of the gold colloidal solutions and all particles, substrates and all. We are able to see the color and do the diagnosis. Now, immunofluorescence. Now, what is fluorescence? Fluorescence is the property of absorbing the light rays of one particular wavelength and emitting the rays of different wavelength. Okay, that we have learned in 12th class also. Fluorescent dyes show up brightly under ultraviolet light as they convert ultraviolet to visible light. This technique is generally used to locate the presence of specific antigen on the cell membrane, and can also be used to detect the antigens present inside the cells. Commonly used fluorescent dye are fluorescent isothiocyanate that emits yellow-green light, and rhodamine B. You should remember some of the fluorescent dyes which are being used. So easy to remember is isocyanate and rhodamine B. Okay. So fluorescents are direct, indirect, immunohistory, and flow cytometry. Now coming to the direct, what happens in direct? Direct is like we do it for the unknown antigen. On the slide, unknown antigen is put. We add the fluorescent label as specific antibody, and due to the fluorescent that combines with the antigen, it presents of UV light, it generates fluorescence, so it is tested positive. In indirect, a known antigen is to be taken. On the slide, we put the patient serum that has antibody. Antigen combines with the antibody, and then we add fluorescent labeled antiglobulin, which in UV UV light that will produce fluorescence. That is also so. That is one main difference between direct and indirect is in direct, unknown antigen has to be taken. In indirect, known antigen has to be taken. In immunohistory technique, it combines specificity of serology with the localized capacity of histology. Okay, so again, the fluorescent thiocyanate and rhodamine, these have to be used, and by combining serology and histology in these two, immunofluorescence helps to visualize antigen antibody reaction in situ. So disadvantage is frequent occurring of non-specific fluorescence in tissues and other materials. So this technique is also not being so used. Like this, we also have peroxidase technique and all. We go to detail, we'll come to, we don't need to go. Now coming to the flow cytometry, it is a device which quantitatively analyzes and separates the cell labeled with fluorescence antibody as they pass through the laser beam and allows the multiparametric analysis of physical and chemical characteristics of up to 1,000 particles, right? And different population of molecules, cells, particles can differentiate it by size and shape using the forward and right angle scattered as specific version of FACS, that is fluorescence activated cell sorter. We are using this for CD4 counting. Okay, so what is FACS? Now you can see in this diagram, we put the sample and there is a sheet fluid also to maintain their flow. We add fluid to the sample, and what happens is that with the help of hydrodynamic focusing, cells passes through the single file. And here is a laser which is being focused, and this laser and focuses on the cells, and with the help of fluorescence emitting stain cell are detected, and forward and side scattered light of all them. So this laser has been thrown, so fluorescent emitting cells have been detected. So this technique is a cell labeled with the specific antibody and tagged with the fluorescent dye is passed to an ultrasound controlled nozzle. So these cells which are stained with fluorescence, they are passed with the ultrasonic controlled nozzle, and this nozzle generates micro droplets and allow one cell to go at a time. You can say one cell is going at a time, and the cell from the nozzle passes through the laser that excites the fluorescence dye, resulting in emission of the light in the specific film. You can see that the laser has been thrown on the cells and fluorescence is being emitted from the stained cell, which is being detected according to the wavelength. And two things will happen during this follow. Laser beam is deflected each time of cell processing, and fluorescent light is emitted. Both these events are recorded by computer and generates plot of recorded data indicating the number of cells on y-axis and fluorescence emitted by the x-axis. Okay, so again this has to be taken in the graph, and using this electromagnetic field, outflow of cells are collected into separate containers. These devices are hence doing two things: detecting the cell with emit fluorescence and collecting the cell separately. So applications are we have to count if we want to count given type of WBC in the sample, and for sort of CD4 and CD8 which we are using. Also for diagnosis and phenotyping of leukemia, we use this. So I think this was it. And if you have any queries, you can always contact me. And I hope now next lecture I'll be taking on the Cisco WebEx application. So let's meet you there. So till then, bye. And if you have any problems, queries, you are free to call me or WhatsApp me. Okay. Thank you.